afternoon. My name is Julian Smith. I'm the Chief Financial Officer of Abzena, and we joined the AIM market of the London Stock Exchange last summer, raising 20 million, and our current market cap is a smidgen under 80 million pounds. We are a provider of services and technology to aid the development of better biopharmaceuticals. I'll explain what I mean about those in a minute. And we do this by utilizing two companies which are part of the group, which we've retained their names because they've got very strong market presence where they operate. So the company's called Antitope and Polytherics. And we've got a mixed revenue model where we're getting service revenue up front with high margins for this part of the business, and then significant upside in the future coming from royalties and milestones as the licensed technologies come to market and we earn royalties. Okay. We also have substantial know-how and patent portfolio that's been built up over the last 12 years. So what I want to do is paint a little bit of the picture of who we are, where we are, and what we do. So first of all, our shareholder base. So our shareholder base, our largest single shareholder is Invesco. We meet with Mark Barnett at Invesco, and he has been supporting the company for a number of years. Imperial Innovations, who Russ Cummings, Chief Executive of Imperial Innovations, is talking across the corridor at the moment, was a founder investor of the Polytherics part of this business, going back 10 years. And Neil Woodford at Woodford Investment Management is a supporter of the company, and I think we were his first company to turn up to his new offices, and he supported us at the IPO. We also have a number of investors who've supported Polytherics during its growth as a startup company, and in fact, because they don't make the 3% cut, which is on this slide, we also have Mercia, and thanks to Mark Payton, who was on our board for many years as we went through this journey. So this is important because part of the story for any company is about the company you keep, it's about your customers, it's about who your shareholders are, and it's about the management team as well as the technology you're providing. So we are comfortable that we have a stellar shareholder base to support us for the future. Where are we? Basically, we're in Cambridge. Any of you who had met Polytherics in the past would have known that we had an office in Camden. All that science base has now moved onto the Babraham Research Campus, and we now are the largest private company on the Babraham Research Campus with just under 100 employees, most of which are involved with the science. We have a small satellite office doing chemistry work, which arose from the acquisition of a technology a company called Warwick Effect Polymers, which is still at the University of Warwick Science Park. And the lozenges at the bottom I won't dwell on, but it shows the mix of scientific skills which we have in the organization, and they all talk to each other. Most big pharma companies have this spread of skills as talk to each other. So the next thing I wanted to introduce you to is the board. As said, I'm a chartered accountant. There on the top right-hand corner. My experience since getting an engineering degree from Oxford was to go straight to the dark side, where I joined Arthur Andersen. I spent 9 years there and then have been in senior level finance roles in technology companies. My most recent role was to join Imperial Innovations, where I took them public and spent 7.5 years there, raising over 220 million of debt and equity for them. John Burt, our chief exec, he can't be here today, but he is a scientist who also turned to finance and accounting. He has experience in business development and took a startup company that was sold to Wyeth just prior to Wyeth being acquired by Pfizer. And Ken Cunningham, our chairman, was most recently the chief executive of Sky Pharma and has been involved in the biotech industry for many years. With me today is Sally Waterman, who, if you've got any science questions, aim them at her because she will understand them far better than I will. but she has experience in drug development as well as processes and operation and regulatory roles from her past experience. And then Matt Baker, who's the last member of the management team that I'd like to sort of introduce to you, was the founder of Antitope. So he has built from the bottom up a profitable services business, very commercial animal, but very, very well regarded amongst the scientific community in which the Antitope company and brand traded. And then at the bottom we have our non-execs with Peter Grant, who's the current CEO and was the CFO of Sky Pharma, Nigel Pitchford, who's the Chief Investment Officer of Imperial Innovations, Tony Brampton, who was a banker at Cazenove, and Anke Lundemos, who is a seasoned Chief Exec of biotech companies. I dwell on that because it's important for people to understand that this is the team that's running this company. So what do we do? Okay, told you who we are, where we are, and who our shareholders are. What do we do? Well, the first thing we do is, in the service side, we do immunogenicity assessment. Now, it's a bit of a mouthful. What does it mean? When you give someone a biologic product, you inject this into them, a vaccine, you want that to generate a reaction in the immune system. Otherwise, it's a vaccine that doesn't work. When you give someone a drug to cure them of cancer or arthritis, you don't want an immune response, because if you get the immune response, the body rejects the cure itself. This is bad for two reasons. First of all, it's bad because the patients don't recover. Secondly, from the drugs company's perspective, it's bad because the patients switch to a different cure, so you lose revenue. So we have a service business which addresses this. Okay? We can then take this one stage further and help the developer of the drug to re-engineer their product so that it doesn't do this, and this is what we call protein engineering. And we have our own proprietary technology in this space such that when those products come to market, we will take a small royalty. And I'll give you a flavor for some of the candidates that are in the pipeline at the moment. Okay? The other thing we do is, in the middle lozenge, bioconjugation, which is basically, layman's terms, sticking molecules together, right? And this is very important. We have a lot of experience at Polytherics of attaching molecules together and making these molecules work and improving their performance in patients. We're working very closely at the moment in the field of antibody-drug conjugates, which is a very hot space, and I'll come on to that next. Actually, no, I'm going to deal with this slide next. So this slide shows on the bottom axis, the x-axis, the prevalence of the negative immune reaction and immune response as found by patients in the clinic. So if you're on the right-hand side, your patients are going to have to switch off the drugs or they're going to stop working. Okay, the left-hand side is the response you get from our process. What you can see from this is a clear correlation between what is found in the clinic and what our process would predict. Okay, we can't say for certain this will happen, we're just saying it's a lower risk that this will happen. We are the only people that can demonstrate this chart, and our business development people, this is the only chart they show people because the drug developers get this chart. If you're in the bottom left-hand corner, your candidate is going to be fine. If you're in the top right-hand corner, you're going to find people switching. So you need to change, use a different candidate, or re-engineer. Taking those forwards, where we've done the protein engineering and we've managed to secure a royalty on the eventual product, what I'd like to do just briefly is dwell on the top 6 of these candidates. All right. So we have a portfolio of about 30 of these licenses, which are covering products which are being developed in the market by our customers. Okay. 2 of them you'll see are with Gilead. Now, for a small company in Cambridge to manage to extract a royalty out of Gilead, for those of you familiar with this space, is quite hard. Well, we didn't. What happened was we were working with a small biotech. The small biotech company was prepared to give a royalty. That product was successful. The company was acquired by Gilead, and lo and behold, we now have a potential royalty stream which is being invested very heavily in by Gilead, so we're not spending a penny on this, and this is stated by various analysts as two of the indications that they're looking for could be blockbusters on their own. So there's a potential royalty flow here which could start in 3 to 4 years' time of many millions of pounds. And all we have to do is sit and make sure that we keep track on what's going on. We have no other special knowledge of any of these programs other than what's publicly available. But I would say that the chief exec of Gilead is talking about simtuzumab a lot. I now move on to the antibody drug conjugates. So the concept here, the simple concept is you take an antibody, which is used for the treatment of cancer, most significantly. It does have other uses as well, and you glue a poison to it. And therefore you're targeting chemotherapy. So the antibody travels around the bloodstream, effectively inert. When it gets absorbed into the cancer cells, the glue dissolves, the poison is released, and the cancer cells are destroyed. Sounds simple. Actually, the key thing is the linker. It's the glue, because if the glue breaks too soon, you're dropping a very, very toxic poison into the bloodstream where it can do all sorts of damage, which is not the intention. If you make the glue too strong, it gets taken into the cancer cell and it works no better, or even in some cases worse than if it is just the naked antibody. And it's that linking technology where we have our proprietary knowledge of how to do that and retain the stability of the antibody so it doesn't break down in the bloodstream. This is a very hot area in the biotech world. So a couple of graphs here for you. First one I'm going to talk about is the Abzena. This is a drug sold by Seattle Genetics dealing with lymphoma. Now you can see the central line is their antibody drug conjugate, and it shows how the tumor volume in the clinical trial progresses over time. So it's better than the black line, which is without the poison attached to the molecule, but it's nowhere near as good as the green line, which is what the same result, which is the result you would get if you used our linker technology rather than the linker technology that they have used. The right-hand chart is a chart for Kadcyla. Now, Kadcyla has been in the press a lot over the last 6 months because this is the breast cancer drug which NICE is refusing to fund, which is out from Roche. And you can see here, maybe this is the reason why they're refusing to fund it, because you can see the tumor growth on the pink graph is still growing, whereas actually the tumor is completely eliminated by about day 12 if they had used our conjugation technology. So this is quite an exciting area. It's quite an early area. We are in early stage negotiation with a number of different people, and it's quite a secretive area. The pharma companies don't like their names being leaked on this sort of stuff because they like to play their cards and control their news flow. So if I sort of take you through the sort of a summary of what we are doing, we have service income, as explained from the immunogenicity assessment. It does more, though, than just give us the revenues from the service. What it also gives us is an insight, because we're in the market, we're playing, we're going to the conferences, we're meeting the scientists of where the market is going. And this really helps, because with that, you can then look to see where you should be taking the business as you move forward. The future revenue and the really large numbers which will come from that portfolio of licenses. Remembering, we're not spending anything on the R&D on those, so we're not going to be coming back to the market saying we need 100 million to develop a drug to do X or Y. That's our partners' jobs to do that. And they will come from the existing portfolio and from the new technology as exemplified by the ADCs and others. I don't have time to dwell on it all today. And finally, in this space, there are a number of small niche providers who have portfolios of licenses, as I've described, who have small service revenues, but they're not big enough in themselves to excite VC or private equity investment. They're not big enough to float by themselves, but they are an opportunity for us to acquire them and to bring them into the group. So you'll also see a buy and build strategy to grow this company and acquire other companies in this way. I'm conscious we have slides back at the booth that we can talk through and some handouts on numbers. And that's the talk. Thank you.